Have you ever wondered how scientists determine the concentration of certain elements in a given sample? Well, one of the ways they do this is through a process known as flame photometry. This scientific technique is a cornerstone in the field of analytical chemistry, with its roots dating back to the 19th century. At its core, flame photometry is a method that uses the principle of emission spectroscopy. It's like a detective tool for chemists, enabling them to identify and measure the quantity of certain metal ions in a sample. Imagine being able to decode the mysteries of a substance just by observing the color of a flame. Yes, that's the beauty and essence of flame photometry. It's not just about burning things and watching the pretty colors. It's about deciphering the secrets that those colors hold. So, are you ready for a journey of discovery? Dive into the world of flame photometry and discover the magic behind this incredible instrument. The principle of flame photometry is as fascinating as it sounds. Let's dive in and understand what this is all about, shall we? Flame photometry, also known as flame emission spectroscopy, is an analytical technique that is rooted in the principle of atomic emission. Now you might be thinking, what's atomic emission? Well, atomic emission is a process where atoms, when heated, become excited and emit light of a certain wavelength. Think of it this way. When atoms are heated in a flame, they absorb energy and get excited, kind of like us when we've had one too many cups of coffee. This energy causes the electrons in the atom to jump to higher energy levels. But just like us, after the coffee wears off, these electrons can't stay excited forever. They eventually fall back to their original energy levels, and in doing so, they release energy in the form of light. This light is the key to flame photometry. Each element emits light at a unique wavelength when it's heated, which can be measured to identify the element. It's like every element has its own unique light signature. Now, here's where it gets even more interesting. The intensity of the light emitted is directly proportional to the concentration of the element in the sample. In simple terms, the brighter the light, the more of that element is present in the sample. This relationship between light intensity and concentration allows us to not only identify what elements are present in a sample, but also to quantify them. It's like having a detective and a mathematician all rolled into one. So, the light emitted tells us the story of the element's concentration in the sample. From this, we can unravel the mysteries hidden within different substances, whether it's the minerals in our drinking water or the components in a far-off star. Fascinating, isn't it? Now that we understand the principle, let's explore the instrument that makes it all possible. A flame photometer, as you might imagine, is not a simple piece of equipment. It's a complex assembly of parts, each with its unique function, working in harmony to deliver accurate results. Let's start with the sample introduction system. This is where our journey begins. The sample introduction system is responsible for supplying the sample solution to the flame. It usually includes a nebulizer and a spray chamber. The nebulizer converts the liquid sample into an aerosol, which is then carried into the flame by a stream of air or oxygen. The spray chamber, on the other hand, ensures that only the finest droplets reach the flame, contributing to a uniform and stable flame. Next, we have the heart of the flame photometer, the burner. The burner's role is to produce a flame at a constant temperature. It's here where the magic happens. The metal ions in the sample are converted into gaseous atoms and then excited to higher energy levels. This process is known as atomization, and it is crucial for the functioning of flame photometry. Following the burner, we have the monochromator. The monochromator is like the gatekeeper of light. It allows only specific wavelengths of light, corresponding to the element being analyzed, to pass through. This is achieved by utilizing a prism or a diffraction grating. The monochromator essentially filters out all the unwanted light, ensuring that only the light of interest reaches the detector. Finally, the journey of the light ends at the detector. The detector is a sensitive piece of equipment that measures the intensity of light. It converts the light signal into an electrical signal, which is then amplified and displayed as a reading. The intensity of the light is directly proportional to the concentration of the element in the sample, thus allowing us to quantify the element. But a flame photometer is not just a collection of parts. It's a carefully designed system where each component plays a crucial role. The sample introduction system ensures a stable and uniform flame. The burner provides the energy to excite the atoms. The monochromator filters out the unwanted light, and the detector quantifies the light intensity. Each part, in its unique way, contributes to the precise and accurate functioning of the flame photometer. And there you have it. That's how a flame photometer works. It's a fascinating piece of equipment that brings together several scientific principles to deliver accurate results. It's like a symphony orchestra where each musician plays a different instrument, but together they create a beautiful piece of music. In essence, the flame photometer is an orchestra where each part plays its unique role in creating the symphony of light. Now that we know how a flame photometer works, what can we do with it? You may be surprised by just how versatile this tool can be. Let's dive into some of the fascinating applications of flame photometry. In the medical field, flame photometry is a trusted ally. It's used to determine the concentration of certain essential elements in the human body, like sodium, potassium, and calcium. These elements play critical roles in our bodily functions. For example, doctors use flame photometry to measure sodium and potassium levels in patients' blood samples. This helps them diagnose and monitor conditions like kidney disease, heart disease, and electrolyte imbalances. But the reach of flame photometry extends far beyond healthcare. Let's turn our attention to agriculture. Farmers and scientists use flame photometry to analyze soil samples. By determining the concentration of potassium and calcium in the soil, they can assess its health and fertility. This information is crucial for guiding decisions about crop rotation, fertilization, and irrigation. Our next stop is the food industry. Ever wondered how companies ensure their products have the right amount of minerals and nutrients? You guessed it, flame photometry. From checking the sodium content in potato chips to measuring the calcium in dairy products, this technique is a mainstay in food quality control. Finally, flame photometry plays a significant role in environmental studies. It's used to monitor and study the concentration of alkali and alkaline earth metals in various environmental samples. This helps scientists understand pollution levels, track changes in water quality, and even predict the impact of certain environmental policies. As you can see, flame photometry is a powerful tool in many different fields. Its ability to determine the concentration of alkali and alkaline earth metals makes it indispensable in everything from medical diagnoses to environmental conservation. From diagnosing diseases to checking soil health, flame photometry has a multitude of applications. Let's recap what we've learned about flame photometry. It's been an enlightening journey, hasn't it? We've seen how flame photometry works, the equipment involved, and how it's applied in various fields. We started with the principle of flame photometry. We discovered that it's a method used to determine the concentration of certain metal ions among samples. It's all based on the principle that these ions, when in a flame, will emit light at a specific wavelength. This light emission is measured to determine the concentration of the metal ions. The brighter the light, the higher the concentration. Then we delved into the instrumentation of flame photometry. We learned that the key components include a flame source, a sample introduction system, a monochromator, and a detector. The flame source provides the energy needed to excite the metal ions. The sample introduction system delivers the sample into the flame. The monochromator separates the light into its component wavelengths, and the detector measures the intensity of light at each wavelength. We also explored the many applications of flame photometry. In clinical laboratories, it's used for the determination of sodium, potassium, and calcium concentrations in biological samples, which is vital for diagnosing various health conditions. In environmental testing, it helps determine the presence of pollutants. In agriculture, it's used to analyze soil and plant samples to assess nutrient levels. In the realm of research, flame photometry opens up a world of possibilities, offering a reliable way to measure and analyze the elemental composition of various samples. It's a technique that, while rooted in the classic principles of chemistry, is continually being refined and expanded upon to meet the evolving needs of science. So, that's the fascinating world of flame photometry. It's amazing how a flame and light can reveal so much about a sample. It's a testament to the beauty and intricacy of chemistry, a field that continues to unravel the mysteries of the world around us. Subscribe for more because you won't find me later.